Hello, welcome to another episode of Science Talks, a conversation hosted by the University of Arizona's Bio5 Institute. My name is Amy Randall Barber. In many cases where babies are born prematurely, they often have underdeveloped lungs. This can cause breathing problems and neonatal lung diseases that require long-term treatment or even lung transplantation. It's critical for research to be done to provide treatments that can help these premature babies survive so that they can grow up healthy. Today, we are joined by a new Bio5 member, Dr. Vlad Kalinichenko, an internationally renowned research scientist and director at the Phoenix Children's Research Institute at the University of Arizona College of Medicine, Phoenix. Dr. Kalinichenko was recently awarded a $3 million grant from the National Institutes of Health to develop new treatment methods for lung disease in premature infants. Dr. Kalinichenko, thank you so much for joining us today.
Thank you, Amy, and I'm really glad to give this interview to Bio5.
We always like to start every podcast the same with a few fun icebreaker, rapid fire questions that are just meant to be simple and easy to answer. So question one, if you could have dinner with any historical figure, who would it be and why?
So I would actually be honored to have a dinner with Charles Darwin. So Charles Darwin, as we all know, it's a historical figure, and his work on the origin of the species is just outstanding. I enjoy reading it, not just from the scientific perspective, but also from the perspective of the traveler and how Charles comes with this conclusion. So he definitely would be my choice.
Okay. Wow, cool. So if you could have any exotic pet, what would it be?
Yeah, well, it's actually interesting. So I would probably have a liger. So the liger is the hybrid between lion and tiger. So they are very social like lions, but like to swim as tigers. So I hope it's exotic enough. So there is a quite few pets, liger pets worldwide. It's about 280% of them in the United States. So, of course, the problem is the ligers are much bigger than lions or tigers, and they need about 50 pounds of steak per day to satisfy their appetite. So, of course, I cannot afford that, and unfortunate. So I'm never gonna get that.
That sounds really cool. I've seen documentaries on ligers, and they're amazing animals. Okay, final rapid fire question. City life or countryside? Which do you prefer?
Probably neither. So I don't like really inside of the city or far away in countryside. I'm a suburban person, so I like to be close to city, but also have some sort of privacy in my backyard. So that would be my suburban, my answer.
Yes, that makes sense. Right in the middle. So it's always nice for us to hear how our members initially got into science. Would you briefly share your story with us on how you got into science initially?
So I think the key part of my career was my training at the University of Illinois with Robert Costa. Robert Costa is a very famous molecular geneticist who discovered a lot of transcription factors, especially from forkhead box family, and that is very important genes which regulate cell signaling in many cell types. So working with him, I actually became interested in a couple of genes from that family, and they happen to be in the lung, highly expressed. And in my own lab later at the University of Chicago, and that was a Department of Medicine, Pulmonary and Critical Care, I actually take my experience with those transcription factors to work in the lung development, lung regeneration, and injury repair. And later on at Cincinnati Children's Hospital, I was a chair of lung regenerative medicine, kind of with the idea of targeting these important genes in pediatric lung diseases, in particular this neonatal lung diseases, which we've been talking about, and I was getting grant from NIH.
Thank you. Thank you very much for sharing that. And so you were at Cincinnati Children's Hospital Medical Center. How long were you there?
Yeah, well, I was at Cincinnati Children's for 16 years, and
Okay.
that's a long way from being a junior faculty member until professor and endowed chair in lung regenerative medicine. So this is something which is kind of was a key part of my career and solidify my interest in the lung as an organ and also in lung diseases as diseases which needed to be treated in the neonatal period. So later in life, these kids have a good quality of life.
So what was it that you would say eventually brought you to Arizona about a year ago or so?
So I would say it is more like opportunity of growth and build something special. So here in the partnership between University of Arizona, College of Medicine, Phoenix, and Phoenix Children's Hospital, the new institute was created with the idea of bringing pediatric translational research in Phoenix. And I was offered the opportunity to be founding director of that institute. And so this interest and getting something special which could affect the patient care in the pediatric population is my mostly goal and interest to come here in Arizona, despite it's quite hot here, but actually I'm enjoying all other times of the year except summer. And I think it is a really great city to live in.
Yes, so you've been here for a full 12 months coming up here in March, and well, today's March 1st. And so you've gotten to experience all of our seasons, which many are the same.
Yeah, absolutely. And actually, I've been through summer in July where we had record heat and it didn't bother me a bit. Well, we walk a lot on science, so with air conditioning, it doesn't make much difference either.
Right? Yeah, we're lucky to have AC in all of our office buildings and homes. Can you tell me about what you're studying now? And you have an administrative position as the founding director, but you also are still running a lab currently, correct?
Yes, I am, and that requires a lot of effort on my part. And we have several different directions to kind of develop new therapeutic approaches for treatment of neonatal lung diseases. And these directions include identifying new small molecule compounds, so the drug screens for a new small molecule compounds which have bronchiogenic properties, so we can induce formation of capillaries in alveoli in neonatal period. And then we are interested in a gene therapy in which we can deliver bronchiogenic genes into endothelial cells of neonatal lung with high efficiency and precision. We're also interested to develop a cell therapies in which we can isolate endothelial progenitor cells from a variety of sources and actually treat kids with this disease. And that is, of course, needed to be developed all the way from small animals and moving to the clinic. And I also interested to develop artificial lung tissue from pluripotent embryonic stem cells. And that is with the big idea to have that tissue for lung regenerative medicine purposes and even for transplantation.
And as I understand it, your lab was one of the first in the world to use stem cells to generate bioengineered lung tissue. Um, can you kind of tell us about that advancement and what led you to that sort of ahead of the crowd, so to speak?
Yeah, so that is very interesting cell type, pluripotent stem cells. With their discovery and with the Nobel Prize being given to Yamanaka for discovery of that cell type, it's become very interesting the avenue to differentiate embryonic stem cells to different cell types. And lots of people in the world are trying to differentiate embryonic stem cells into respiratory cell types. And they do it either in vitro, in organoids, and there is a lot of research going on now, which is very clinically translationally relevant because the cells can be potentially used for a variety of purposes. So my lab is interested in actually using embryonic stem cells to create lung tissue using embryos as a bioreactor. So in this case, we are not using soluble factors or artificial scaffolds. We are using Mother Nature to guide us how to make embryonic stem cells from how to make lung tissue from embryonic stem cells. And with that, the interesting method called blastocyst complementation, in which you can inject embryonic stem cells into early embryos called blastocysts. And if these blastocysts are genetically modified through CRISPR-Cas9 technology to remove important morphogenetic genes, which, for example, drive lung development, in this case, injected donor pluripotent embryonic stem cells would go to lung field and build the whole lung tissue. And that can be done actually between species. So my lab was one of the first in the world to generate lung tissue in the mouse from mouse embryonic stem cells. And we now continue these efforts and have a very strong study which is showing that we can generate mouse lung in the rat. And that mouse lung is pretty truly mouse cells, but resides in the rat, and that in the embryos called interspecies chimeras. So that if that technology can be applied to humans, that there is an avenue of generating lung for transplantation for people who would need lung transplant in the future. Unfortunately, right now, because of different reasons, ethical, political, religious, and other things, this experiment cannot be done with human embryonic stem cells and human embryos. But I think the technology is not there yet. It needs to be refined and adjusted first in small animals before we actually can even propose to do something like that with the human embryonic stem cells.
So I mean, if you could have a guess, how many years are we talking before we even get there? Do you think it would be, 10, 20 years?
Well, I think that is a long way to go. Basically, there are several labs in the world which started working with very early embryos using human material. And of course, there is a lot of barriers with that, but that is very early, the effect of stem cells on development of the organs in the very early time points. So whether it's gonna go later, we basically don't know. It's not up to us to.
Yeah.
That's probably there's a variety of laws needs to be actually modified to let us do it, and of course needs to be very heavily controlled, and just to make sure that no ethical or political or religious reasons actually change that experiments. And basically, I would estimate that it could be 10 years, it can be longer, and it starts from small animals first and then goes to big animal, and finally, if that everything is works well, then even after that, the human experiments could be considered after extensive review and approvals, and that would be on the highest level approval of such experiments.
Yes, the ultimate success in in your work.
Yeah, it's true, and it's not about lung transplantation. So basically, any organ can be generated like that with a combination of this blastocyst complementation and CRISPR-Cas technology. And of course, we would need to have some large animals, which probably should be genetically modified and remove some of the genes which would affect immune responses and engraftment of embryonic stem cells. So it's a lot of things we don't know. We need to prevent integration of embryonic stem cells into the brain tissue, sensory organs, reproductive organs, because that would be main concerns for potential human use. But I'm very optimistic in this direction. So I think in years to come, we would generate transplantable organs somewhere on the farm, like same way we are growing steaks. And that would be a very big break for human beings, for especially people who are waiting for transplantable organs.
Absolutely. Wow. Thank you for that explanation. So just kind of going back a little bit, can we talk about just neonatal lung disease? And just can you explain a little bit about what that means? These babies are born prematurely, and their lungs are underdeveloped. Are there particular diseases that they end up with that would kind of lead to a transplantation or treatment?
Yeah, there are many neonatal lung diseases which are detrimental for human health. For example, there are genetic diseases in which several important genes mutated, and these babies are born with the lungs which are not capable to perform respiration. And then among these diseases are alveolar capillary dysplasia with misalignment of pulmonary veins, the acinal dysplasia with mutation in TBX4. There are also surfactant abnormalities with mutations in surfactant genes and other genes related to surfactant homeostasis. But in addition to that genetic diseases, we also see patients with lung diseases which are born prematurely. So we can rescue babies who are 22, 23, we deliver 22, 23 weeks of gestation. And these kids are born with the lung which is far away from being perfect for breathing. And these kids are needed to undergo a variety of therapies, including supplemental oxygen to basically deliver the oxygen into peripheral blood, and this oxygen delivery actually causing injury, and that injury aiding on prematurity, and that results in actually lung born with big holes, which in adults would call emphysema, but in kids it's called bronchopulmonary dysplasia. So, and there are cases of congenital diaphragmal hernia in which diaphragm is not developed correctly, and that results in abdominal organs pressing on the lung, and that causing compression, and the lung was not formed correctly. This particular condition can be treated through surgery, but because lung been compressed in utero, we have big consequences of this compression, and these kids have very underdeveloped lung and have a variety of conditions, including pulmonary hypertension later in life.
Thank you very much for explaining that. So I'm going to forward ahead to your role as director of the Phoenix Children's Research Institute at the University of Arizona College of Medicine, Phoenix. Can you tell us a little bit about your role there? I was reading a little bit that they had some goals kind of set up for you right away, one of which was met. We got a grant in, a very large, wonderful grant. Can you tell us a little bit more about your role there?
Yeah, so as a director of Phoenix Children's Research Institute, I am in charge of recruiting strong investigators which are interested in developing new technologies which can help for variety of pediatric conditions, not just lung, but any other organ. We have a very strong clinical operations and Phoenix Children's Hospital, and of course bringing new technologies and bringing new science, which is would be a kind of the point of developing new treatments for diseases is very important for us, and that would result in adding additional clinical trials. So I'm now trying to attract here to University of Arizona Phoenix strong investigator with outstanding translational programs which are related to children's health. So that is the idea, is a multi-year recruitment effort, and which would hopefully bring Phoenix Children's in the best children's hospital, not just in the United States, but also in the world.
Yeah. Wow. Well, that is a big commitment, and I have no doubt that you will be right there meeting all of those goals. So what's I know that you just got into this position. We do usually like to ask what's next for you, but it sounds like what's next for you is seeing this through it and getting the recruitments and building up the hospital. Is there anything else that you had in mind for your future?
So, of course, for the recruitment, that is a primary goal, but also also from my scientific interests, I would like to move some of our discoveries, which we already done, into clinical trials. And for that, we would need partnerships with biotech companies, which would help us to develop the nanoparticle delivery vehicles, which basically we discovered in our lab specific nanoparticle delivery vehicles which can target endothelial cells, but of course to produce it on a large scale and with good manufacture practice, and that is it's something which is traditionally been used to characterize a human grade products. So that is requires actually biotech company to produce it and go with all additional tests, and that's needed for FDA to consider any potential human use. So basically, moving our products and gene therapies, cell therapies, and newly discovered drugs to potential clinical trials, that would be my goal from scientific point of view, and that is highly relevant to what we try to accomplish at the Phoenix Children's Research Institute. And of course, Phoenix Children's Research Institute, not about lungs, but about every single pediatric disease which we can potentially treat with new therapeutic approaches. So that's why we are not focusing only on one organ, but trying to expand our interest into many different pediatric diseases.
Wonderful, thank you. So for our final question, what is your why? What keeps you going in all of this? What gets you up in the morning?
Well, I think it's about goal, what we try to achieve in a life. So in my case, I am just trying to do my job, and from different perspective, from science, I know my goals, and that is different scientific directions, and we are trying to achieve new breakthroughs in different directions and publish research manuscripts, which also results in obtaining new funding. From the recruitment purposes, meet new people, have new conversation, and try to pursue them to go to University of Arizona College of Medicine, Phoenix, and Phoenix Children's Hospital. That's a long-term commitment to build something special, and I hope to achieve that in a few years down the road.
Absolutely. Thank you. That's wonderful. Well, thank you so much for being with us today. We really appreciate having you as our guest, and we thank you for answering our questions and having a conversation with us today, and taking the time out for that.
Thank you, Amy, and I appreciate you having time with me.
Thanks to our listeners for tuning in to another episode of Science Talks. Continue the conversation with us next time as we learn more about the amazing science happening at the University of Arizona's Bio 5 Institute.